in this particular segment, you try to understand exactly what is happening at the molecular level while two DNAs are recombining. And here we would know the different factors, different proteins that are involved in the process, and other mediators of the process, and also different types of biological significance of recombination, as well as various application of recombination. So let us start from the very beginning of the process. So here, just let me share my PPT. So at the initial segment, we have to discuss, of course, with homologous recombination. Now, homologous recombination, it is probably the most significant recombination in terms of generation of genomic variation. Okay, but other than homologous recombination, there are other types of recombination also, and that's mechanistically those are quite distinct. So what is homologous recombination? For that, we have to know first what is homologous sequences. So homologous sequences are sequences which are not really identical. That means both the sequences are not absolutely 100% match, but they are similar. So there is a certain level of identity and certain level of similarity between the sequences. These homologous sequences are actually exploited in homologous recombination. That's why actually the homologous chromosomes get involved into exchange of their DNA segments. So homologous recombination, it is recombination based on DNA sequence homology. There are other recombination like site-specific recombination, which depends on the presence of specific DNA sequences, not homologous. So specific DNA sequences in one DNA and another DNA sequences, another DNA can actually trigger a recombination that we call site-specific recombination. And then there is a completely different kind of recombination that we call transposition, where specific enzymes are involved to cut a piece of DNA and then to join it with another piece of DNA. So that is called transposition. So these two are completely distinct mechanistically from homologous recombination, which is absolutely dependent on sequence homology between two DNA. So the fundamental chemistry of homologous recombination, it is dependent on, of course, homology, but if you look into the process, it's basically nicking of a DNA, then exchange of one strand between two DNA molecules, and subsequently branch migration and resolution and sealing of the nicks that are there. So we will get into the process when we will discuss about the process, its intricacies and other detailings. We will get a better look of all of these processes. Now, homologous recombination, as I mentioned to you, that it occurs, as I mentioned to you, that it occurs between two homologous DNA sequences, right? So ABC and ABC, as it looks that they are similar sequences. So if if when they recombine to produce some recombinant DNA, we call it homologous recombination. But if such event occurs between two non-homologous sequences, we would call it non-homologous or illegitimate recombination. Then there are site-specific recombination, as I mentioned. So site-specific means there is specific sites between the two DNA, like we know while integrating the lambda phage, integrating into the E. coli DNA. The lambda phage uses its sequence called ATP, and it recombines it with a sequence in the E. coli genome ATB, and thereby lambda integrates. A specific enzyme is involved here, which is called lambda integrase, but it depends on presence of the sites at P and at B. Then there is replicative or non-replicative transposition, where a segment of DNA can jump into another part, and this can be through replication or this can be through simple cut and paste, and this is mediated by a group of a family of enzyme that is called transposase. So these are different types of recombination. Homologous recombination, as we have already discussed quite a few times, that it is basically pairing of homologous DNA. In case of meiosis, pairing of homologous chromosome. Then a chiasma formation, which actually is the exchange of specific segments of DNA or strands of DNA, followed by resolution of the chiasma, which results in the recombinant chromosomes. Now, in a DNA, between two DNAs, there can be a single crossover and there can be a double crossover. So, single crossover means a single recombination event initiating at one particular loci, and double crossover means two independent recombination events in two different loci. So when a DNA is crossing over and a single crossover event is there, it would result recombinant DNA, a pair of recombinant DNA like this. So yellow, green, and here you have the recombinant segments. When there is double crossover between two DNA segments, there is exchange of a part of DNA. So here you can see in the middle, the parts are exchanged, like green and blue, those parts are exchanged. So that is the outcome of single crossover and double crossover. Now, in any eukaryotic or in any prokaryotic genome, there are some sequences which are called repetitive DNA sequences. That means they are similar sequences coming more than one times, and most of the time these sequences are located quite close or at certain distance. This kind of sequences can be found out, and these sequences can be a direct repeat. So if you consider this as 5' end, and if this sequence looks like ATCGG, so on and so forth, and if after a certain distance the same there is another sequence which is absolutely similar, ATCG, then this will be called a direct repeat of the first sequence. So these two are direct repeats. Similarly, repeats can be inverted repeats also. So if this looks like ATG whatever, then at a certain point, if you get a sequence which looks like just the sequence which you would read from the 3.5' end, that means just from the opposite direction, if you read, you would get the same sequence. So this is called inverted repeat. Like here you can see this is ATC, GGC, something like that. So this direct and if inverted repeats, those are there, then there can be intramolecular recombination. And what is the outcome? The intramolecular recombination between direct repeats leads to deletion of the segment of DNA that is in between the two repeats. So actually the DNA loops, then the two DNA sequences aligns, and then the recombination occurs, and that results in deletion of the sequence, deletion of the stretch of DNA that is in between the two repeats. Similarly, for indirect repeats, the DNA loops like that. You can see if the DNA just loops like this and comes in this direction, you will find out that those two DNA, these two, the inverted repeats will be able to align with one another. And when it happens, then what will happen? Then homologous recombination between them would result a reversal of the direction of the internal sequence. The sequence that is in between the two indirect repeats, there will be flipping of the sequence. Okay, so the outcome of intramolecular recombination between direct and indirect repeat is quite different. Now we have to discuss about different models for recombination as it was proposed since the day we tried to explain the mechanisms of recombination. Now, Holiday model, this was proposed by Holiday long back. This model involves alignment of two DNA, then formation of a nick at the same position or some position which is nearby, and then strand exchange. So if you consider here the pink and blue DNA, there is a strand exchange between the two DNA. So the blue DNA is getting into the pink one, and the pink DNA is getting into the blue one. And subsequently, this model proposes that immediate after that there will be sealing of these nicks. So this pink blue DNA will be joined, and this blue and pink DNA will be joined, which will form a kind of branch between the two DNA. And subsequently, there will be a branch migration. So this branch will migrate, and more the branch will migrate, more strand exchange will occur. So ultimately, it will migrate at a certain pace, 50 base pair per second or something. And ultimately, it will stop at a place quite distant from where it began. And in between, and it will form a structure that is called Holiday junction. So since it is these nicks are sealed and the branch is migrating, you can call that Holiday junction is formed. This Holiday junction will resolve after a certain time. So after sufficient amount of strand is exchanged, there has to be resolution of the Holiday junction. Now, how this Holiday junction look like? If we look it into, if we look at this DNA from a different angle, it would look like this. And if we just turn this DNA around, it would look like this. So we are not changing the DNA conformation, but actually the DNA Holiday junction would look like this particular conformation. Now you can nick this DNA horizontally or vertically. There is some endonuclease that would come, and that can nick this DNA either horizontally and vertically. And what is the outcome? If it nicks it horizontally, then this blue DNA will come out with this small pink stretch, and this pink DNA will come out with this small blue stretch. We call it patched heteroduplex. Similarly, if you cut it vertically, then this blue segment of DNA, double-stranded DNA, will come out with another double-stranded segment of pink DNA. Similarly, this blue DNA will come out with another double-stranded segment of pink DNA. So this will result in a spliced heteroduplex where a certain number of certain amount of double-stranded DNA from both the DNA is present over here. But for patched heteroduplex, only a strand, a small stretch of strand is present in the recombinant DNA, that is the contribution of the other DNA while recombining. In electron microscope, when the Holiday junction was observed, it looked like this. So you can see there is a bubble here, and these four are the four DNA strands. So it is typically this kind of structure which is observed under electron microscope. Now we have to understand what is strand exchange. Now, if you put double-stranded DNA in solution, that does not necessarily mean that all the DNA in the solution will remain as double-stranded. So there is a certain dynamics. The dynamics of the strands being denatured and being renatured, or the hydrogen bonds that are in between, those are kind of their formation, their disruption. There is a certain kind of dynamics. Although in a solution of double-stranded DNA, majority of the population will be double-stranded, but there will be a certain population which will be single-stranded also. Now, if you put a single-stranded DNA, which is not there, but which can complement with any one of these DNA, what this DNA will do, this DNA will try to base pair with one of these DNA. Okay, because as I mentioned, this is not kind of stable as covalent bond. So the incoming DNA or the foreign DNA or the other DNA that can kind of invade this double strand, and it can form base pair with any one of these, displacing the other strand. So in the holiday model, actually this particular concept is exploited where during branch migration, because these two DNAs are homologous, so the DNA that is invading this should be able to base pair with the other strand. Similarly, for the other DNA, that is also true. The invading strand will be able to base pair with the other strand. So because most of the DNA is similar, there will be some mismatches that will not affect this whole base pairing and stability of the whole system. So the branch can migrate. Therefore, this understanding the strand invasion is very important. So that means if you have complementary DNA, then it can displace a pre-existing double-stranded DNA. Now we would check a better depiction of holiday model. Here you can see A, B, C. These are the three markers, three genes. So in the gray DNA, they are shown as capital A, capital B, capital C. And in the blue DNA, they are shown as small a, small b, small c. Now, this is the point, this block where the both of the DNAs are nicked at one of the strands. And then subsequently there is sealing of the nicks, I mean the exchange of strands, sealing of the nicks and formation of the holiday junction. Subsequently, the branch will migrate. So if the holiday junction is initiated at a loci which is in between A and B gene, it can migrate at a position which is in between B and C genes. Now what will happen? As I mentioned before, that the holiday junction, this will resolve. So this is basically the holiday junction. And when the strand exchange actually occurred, you can see that part of this gray DNA is now base paired with part of the blue DNA, and same for the other one. And here you can see in this segment, one of the B allele is capital B, another one of the B allele is small b. So that indicates that the strand exchange actually resulted in recombination. Now, after this holiday junction formation, if you this particular structure that will form as we mentioned before, so this structure will be resolved by some enzyme. And as I mentioned that when the DNA will be cut vertically, then what would you see? You will see that in one of the recombinant DNA, there will be this for the top strand, there will be a long gray patch with capital A, capital B as the allele, while for the C allele it's small c, and for the other strand there will be a long blue strand with small b, small c as the allele and capital A as the other allele. So similar kind of allelic organization or distribution will be seen in the other DNA, and this would result in a spliced heteroduplex formation. So vertical cut, spliced heteroduplex formation. Then if you cut it horizontally, then it would form a patch heteroduplex. So here you can see that the top gray strand is unaltered, capital A, capital B, capital C. The bottom strand is a recombinant where it is capital A, small b, and capital C. So gray, blue, and gray. Same for the other DNA. The lower strand is unaltered, A, B, C, small a, small b, small c. And the top strand for the lower DNA, it's capital A, it's small a, capital B, and small c. Blue, gray, blue. So these are patched heteroduplex. Ideally, because this cut is random, or presumably this cut is random, so there is 50% chance of patched and spliced heteroduplex formation. So probabilistically, in a recombination event, it can be a patched, it can be spliced heteroduplex. This is what the model. This is the model for recombination that was proposed by Holiday. Now, after that, when we came to know a little bit more about DNA biology, other models are also proposed, and those models are associated with DNA breakage. Particularly, these models significantly described how DNA damages are repaired, and also these models provided us that how recombination can occur where there is single-stranded nicks or there is a big deletion in one of the DNA strands. So, as Messelson and Redding model, it proposes that recombination can even occur if a DNA is nicked at a single strand of only one DNA. So here, red and blue, these are the two DNA that are recombining. So there will be a nick at a certain position, and when there is a nick, this DNA can start replicating. So this DNA will start getting copied by some DNA polymerase, displacing the other strand. And this other strand, as I mentioned, strand invasion, would invade the blue DNA because these two DNAs are homologous. So this invading fragment will be able to find out complementary sequences in the blue DNA, and these would result formation of a loop because the red DNA is invading. So the blue DNA will be denatured and it will form a loop. Now, subsequently, there will be this D loop will be removed. So what will happen? Okay, that part we will discuss later on. Let us discuss the other one. So another model which proposes that there can be two nicks on one DNA which can form a large deletion, and this deletion can be repaired when its homologous DNA can invade here. So the homologous DNA can invade, it can be spared with a single strand. So of course, what this will do, this will actually allow a gap in the top DNA which can be repaired by some, which can be actually replicated by some DNA polymerase. So here you can see this top DNA, this can be copied by some DNA. So in this particular model, the displacement loop that will be removed, and of course the top DNA was copied, so this will result in this structure. According to this model, the top DNA will be replicated or will be actually polymerized, and then this will form this particular structure. So subsequently what will happen, there will be migration of this branch, so the strand exchange might be extended further, and that would lead to formation of some holiday junction over here, and then this junction will be resolved as we discussed before. So that would result into some recombinant DNA. So both Messelson and Redding model actually explains how recombination can also occur. So Holiday, Messelson and Redding model, these are basically two major models. Then there is another model which is called double strand break repair model. So in this model, actually, this model was proposed when people understood how double strand breaks are repaired. So sometime during the exposure to X-ray or some other DNA damaging agent, double strand breaks can be induced, and so this might result into recombination. So these double strand breaks, those will be recognized by some factor which will process these double strand breaks to actually generate some DNA with some extended single strand. So these are sort of cohesive ends with extended single strand, 3' end extended. These extended 3' ends would invade its homologous DNA, and thereby it will form some D loop. Now, this D loop sequence, this looped DNA sequence would be complementary to the sequence of other DNA. So when the D loop would extend, it would serve as a template for the top DNA so that some new DNA can be synthesized. Similarly, within the D loop, some new DNA will be synthesized because that's how the D loop will be extending. So both polymerization would allow extension of the D loop, and in this way, from this double strand break point, actually longer strand exchange will occur. So ultimately what would happen, these will extend till that, and it will be this particular nick of the gray DNA will be sealed with the orange DNA, and here this particular nick of the gray DNA will be sealed with this orange DNA will be sealed with this gray DNA. So ultimately there will be two junction points which will be resolved to generate a recombinant DNA, and which will actually repair the double strand break. So this is how double strand break repair model proposed the mechanism of synthesis dependent strand annealing. It's one of the simpler version of this double strand break repair model. So here, as you can see that here, from these overhangs of these double strand breaks, what would happen that here, as we are mentioning, that in the displacement loop, the complementary DNA is synthesized, and in this particular model, from this complementary DNA after its synthesis, it it is released from this D loop. So the lower DNA is reannealed, and this released DNA links with the other end of the fragmented DNA, and after that the second strand is the top strand is synthesized, so and then ligated to form the completely repaired RNA DNA. So this is basically a model which also explains how double strand break repair can be accomplished by synthesis dependent strand annealing. So both of these models are valid models for recombination in double strand breaks. Now, we would like to start our discussion on components of homologous recombination, and this will be an elaborated discussion. So maybe in our next segment, we will discuss about these components of homologous recombination.